A very good morning to everyone. And on behalf of PKC, Pune Knowledge Cluster, I welcome you all to the second session for this particular course. And may I now request Professor Sujata Bhargav to introduce the speaker.
Thank you, Prachi. It gives me great pleasure to introduce Dr. Bharat Char to all of you. He is the Chief Science Officer at Maharashtra Private Limited, which is Maharashtra Hybrid Company Private Limited. This company deals with R&D, production, processing, and marketing of seeds of different crops like cotton, paddy, wheat, bajra, jawar, as well as some vegetable crops. And Bharat Char is responsible for all the R&D and regulatory programs at this company. Regarding his educational background, he did his PhD in biochemistry and molecular biology at the University of Southern California. And then he did his postdoc at the University of California at Berkeley in plant molecular genetics. So he has been in the academia long ago, and then he has moved to a place, a company, which is very well known in India, and I think even the rest of the world, as a major seed company, which is coming up with very interesting products. I request Dr. Char to start his talk now.
Thank you, Sujata. Just share my screen here. Yeah, so good morning, everyone. Thank you, Professor Sujata and Professor Vidya Gupta for this opportunity. It's great to speak to you this morning. What I thought I'd do is touch on a few topics. The title is kind of broad, so it's molecular approaches. But I thought I'd like to give you an industry perspective where I can as to how we approach some of these techniques and ways to improve breeding. So ultimately, that's our goal. So I'm going to start with a very short overview, like a minute I'll spend on the company. Just so those of you who are not familiar with Mahiko get an idea. So we are now under the umbrella name of Mahiko Grow. We are a group of companies, and it was founded by our founder chairman, Dr. Barwale, in 1964. So I think the reason I'm showing this slide is, one is he's a World Food Prize winner, and the other is that he encouraged R&D, from the outset, and that legacy continues today. And that's most of what I'll talk about is, the seeds were sown literally by him. So we have some firsts, since we started as an improved seed company. Dr. Barwale first started with okra seeds and just improved okra seeds from the Pusa Institute, and then went on to develop hybrids. And we have several firsts in number of crops back in the 60s, 70s, 80s. And we are known quite widely for introducing BT cotton into the country in 2002, and then subsequently the second generation BT cotton in 2006. Now, I'm not going to speak about GM today. We've developed a lot of products which are pretty much on the shelf because of regulatory issues. But BT cotton in India, of course, has been around for close to now 2 decades. And BT brinjal has been released in Bangladesh. It was released back in 2014 and doing quite well there, and possibly in the Philippines this year. So, but I'm not going to speak about those. I'll speak more about the core topic today, which is breeding and techniques that go into breeding. This is our research center, and I'm sitting right there today. So we are, of course, a company that services farmers. So small farmers are our primary customer, and everything comes from that. So all our R&D goals, product development goals are attuned to the small and marginal farmer, and whether it's in terms of productivity, it's in terms of economic return, nutritional value, all these things we are driven by our overall mission of farmer first. Today, what I'm going to talk about is here on this slide, on the left side. So I'm going to talk a bit about molecular breeding because it's the primary tool we use for enhancing breeding through our R&D. But I'll also talk about predictive breeding, which is a little bit newer topic. It's come to the fore in the last 2 to 3 years, and I'll give a little bit of overview on what we are trying to do there. And then moving on to new breeding techniques. These are, of course, gene editing, genome editing techniques. I'll talk about what we have done so far in that. And finally, I'll close out with some discussion on digital agriculture, which is kind of a buzz topic now. And we have made some efforts in it, primarily to support our breeders, and I'd like to talk about that. It's not strictly molecular, but it's going to play a larger and larger role as we go forward. So to get into the discussion, so first we'll talk about molecular breeding. And, of course, phenotypic and genotypic variability are the keys for improving any crop. So when we start breeding or trying to support our breeders, give them an edge, we look for genotypic variability, and I'll talk about that in more detail. And then the markers act as, it could be diagnostics as well as a prognostic or a predictive tool. And these are the 2 areas I'll cover today. So the way we are set up is that we look at our technologies in terms of what is our strength in genomics in a particular crop, what markers do we have, what IT or bioinformatics support we have, and of course the data analysis support is required. And that we use along with our breeding teams to the data that they generate. The genotypic data, of course, comes from the lab, but the phenotypic data comes from our multi-season testing, our product development process, which goes from small yield trials all the way to multi-location trials, multi-year, multi-season trials. So all that phenotypic data is what we try to utilize along with the genotypic data. And finally, when you come to commercialization, and the product reaches that market demo stage, all these things have to have fed in correctly and given the breeder the right results that he can make a decision as to what to advance for commercialization. So all these so-called back-end things play a very large role when you come down to commercialization. So we have to be very attuned to what the market wants. So here, just as an overview, we work on a wide, row crops as well as vegetable crops. This slide just gives a snapshot of the areas we work in for the row crops, so rice, wheat, and cotton. And essentially, there are common practices, obviously, across the crops. We do new line development, new hybrid development. And of course, at the same time, we're developing markers on the lab side. And in the case of rice and wheat, we have also a set of traits that we're interested in. And these are very broad. All rice breeders work on this. It goes from BLB, BPH, blast, all the major pests and diseases, and the traits that are important to the breeder, and finally the grain quality as well, milos, and so on. On the wheat side, rusts are a major issue, and I'll talk a little bit on wheat again later, just to give an example of how we approach the genotypic and phenotypic data collection. So we do a lot of backcross breeding, as you might imagine, any breeding program, and so we have a set of markers for every crop. It's a conventional approach in terms of using markers. There's nothing fancy there. It's just a season by season cyclic effort, and I'll talk a little bit about that. But essentially, we are trying to shorten the breeding cycle by using markers at any stage that we can. So we, instead of doing 6 back crosses, we try to do 3 or 4 to get the breeder to his or her destination quicker. And it's a very routine kind of cyclical activity every season. And this is just a snapshot of cotton off-season plot in Karnataka, which we use for so we can do 2 seasons of cotton in a year. A bit staggered, but we can manage to do 2 seasons. And the samples come from the lab, and this is representative of all crops. The samples, the crosses are made by the breeder. They come to the lab. We have a Tecan robot system to process do the PCRs, and we run it on here. Example is a K-bio system where we genotype. So we have, if you look at all the crops we're actively doing, it's rice, wheat, cotton, chilies, okra, and the vegetable side as well. So it's a large effort, and the 2 seasons, Kharif and Rabi, are very rushed because all the samples arrive at similar times and have to be processed so breeders can make their selection. In terms of the approach to looking at a crop, in our germplasm bank we would have a certain collection, and within that collection we would have some lines that are phenotypically characterized over the years by the breeder, some that are genotypically characterized by the lab, and the lab activity is a more recent activity, and therefore not all the lines are genotypically characterized. But within this whole sort of set of lines, we have a code set for each crop, which we utilize for our elite breeding program. So it's sort of like it's like a Venn diagram, but a subset of that is used for our current breeding efforts. And then at the same time, while the genotyping is going on, we do field phenotyping over years and different locations, multi-location. The trait phenotyping, the traits of interest in whatever crop, rice, wheat, etc. And then looking at specific breeding values such as general combining ability, specific combining ability, since we work with hybrids most of the time. And then wherever we can, of course, we utilize platforms, phenomics, metabolomics, and proteomics. But I would say 90% of our activity is restricted to traditional genomics at the moment. This is an example of one crop, okra. You can see from the initials. So we do a basic genetic diversity study in any crop when we start out doing the molecular marker or data characterization, is to get an idea how diverse the crop is. Sometimes you get surprises. Sometimes they have very narrow genetic base. Sometimes it's a decent spread. But this is the starting point, and we've applied this across all the crops. And then the core set that I talked about, the elite lines, the crosses between them are made, and then the genotypic information is generated for those crosses. And they go into 2 different programs. One is line development, which is, for example, for developing parental lines. And the other side of it is hybrid development for new products. I'm going to this little quickly because there are a lot of slides to cover, but I'm happy to come back and talk about some of these later in the question answer session. In terms of specific traits, I mentioned rice and wheat. But across every crop, there would be biotic, abiotic, grain quality parameters. In some cases, fertility restoration for hybrids, and these are built into each of these programs at screening stages at every season for selection. We also work with transgenic traits in cotton. So, for example, I mentioned Bolgard cotton. So there is for molecular quality assurance, there has to be a certain standard of trait purity and trait presence. So we do that for cotton. So it would be for the 2 genes that are in cotton. We also do a lot of zygosity studies. So to help the breeder, we have developed studies which tell us which of these traits is in a. I'm talking about the GM traits. They are in either the heterozygous or homozygous condition. So that saves a lot of time for the breeder, and they can pick the plants right at the early stage, so that they don't have to do a segregation analysis later. And this is achievable because we have flanking sequence from the insertion of the transgenic trait. But this is a routine activity that is integrated into our screening now for that particular crop. And then, of course, for these trait integrations, whether they are transgenic or conventional traits, we do integration using backcross breeding, and there are many considerations there when we work with particular traits in a particular crop. So population sizes, number of generations, number of markers, all these have to be optimized for a particular crop and trait. The molecular breeding team also does purity testing, so that this supports our QA activities. So whether it's parental purity, hybrid purity, sometimes the grow out testing team does not have enough time to complete the grow out test. That's what GOT is. So we can use markers. It's more expensive, but it gives a result in a much shorter time. And so these are some of the examples we use for supporting the QA team. Now I'll spend a little time on wheat, just as an example of, just to give a little bit of a more specific idea of how we approach things. So in our wheat molecular breeding program, we have basically these 3 or 4 goals. The line improvement for parental line development, then trait integration, and hybrid development. Now, on top of this is a layer of introducing the specific traits that we're interested in. Now, this table just shows the traits, but we have a detailed set of each of these markers for each of these traits. For example, stem rust, leaf rust, stripe rust, or cytoplasmic related markers. These are incorporated at every stage in the breeding. So it forms a second layer of markers which go along with our background selection. And in wheat, we chose to do a genome by sequencing analysis. And the goal was, and this is leading up to the next topic I'll just touch on, which is predictive breeding. So, where is the sort of the science headed? We want to be in a position where not only do we have genotypic and phenotypic information, but we also want to use that information in a predictive manner. So what I talked about prognostic, because these are all very resource intensive kind of activities from a marker perspective. The cost is high, instrumentation is expensive, and what we want to do is optimize the activities such that we can reduce the number of crosses that the breeder makes that need to be analyzed. So it's a very sort of simple idea where you can actually predict the best crosses using historical data. You can bring down the crosses to say 10% of what you would have traditionally done. And so in wheat, what we did was we generated all this sequence data by sequencing some 300 lines, something like that. And then we generated all these SNPs, which would be used in our predictive breeding program. So not only does it give you a genetic diversity picture, so that you can use that, as I mentioned earlier, that is one of the basic things we want, is to have an idea of the genetic diversity to start with, but it's also usable in predictive breeding, as I talk about. So so moving on to that topic, so what is predictive breeding? So just like somebody has, I think there's a line drawn on my screen, but I think it's okay. So just like we have predictions for weather, the financial markets, projections in business, etc. The whole field of molecular breeding is moving towards predictive breeding. And there have been lots of examples around, but as of today, it's still a work in progress. And many of these things I talk about are works in progress. But here, so what is predictive breeding? Essentially, you utilize the historical data from your crop. So whether it's so you can take any crop like rice. You have lots of multi-season data, multi-location data over the years as we've developed our products as a company or any breeding program. And so that historical data is very valuable because it gives you a lot of very precise performance data for those lines. And then you have to merge that with the genotypic data and use a statistical model to and an algorithm to predict what would be the best crosses within your germplasm. So just to go into a little more detail, so we can break this up into different components. So for example, you're starting with whatever you have already, the data that you have already collected can be designated as a training set, because that's what you use to try and make predictions. So it's a historical trial data. It could be lines, it could be hybrids. And you have all the pedigree information from on those lines. And what is done is then you put a high density set of markers on those lines. So you analyze those lines, those training sets, that training set using a high density set of markers, so that you have that picture superimposed onto your performance data. The statistical models are usually they are proprietary now. So many companies offer ways to run this information through an algorithm. And I'm not an expert on it, so I can't really discuss it. But all I say is that they are available now. So even academic groups and industry groups, companies are working on this. And using this data, you address a target set. So the target set is your phenotypes, new lines. And so using the marker information plus the performance information, you can come up with a best set of crosses that could be made. And this is what we are in the process of validating. So it's not a simple process, because it takes time to develop both things. So whether on the genotyping side, do you have a complete set? On the phenotyping side, do you have a complete set? Only then is it worthwhile to spend the time in doing this. So this is where the field is heading, and it's not yet in a position where you can make predictions very confidently. It's still a work in progress, but a very exciting area for molecular breeders, because this gives a chance to do predictions and validate it in the field. I think that's where the greatest impact will be, obviously. Now, just to round out this part of the talk, all this needs some support in terms of information management systems. So the minimum that you require is a laboratory information management system, where you have all the basic germplasm data on your lines, the passport data, what we call characterization data, and the marker data. And then you need a breeding platform. So we use one which is called Phenome, and essentially it manages all the breeding information, the pedigree, the crossing, the data collection, and so on. And using these two, using the breeding platform, you come to our predictions. And so we are halfway there. We have the two components on the left side, and we're still working out the predictions. So I'm going to change gears here and move on to gene editing. Let's see how much time I have. About halfway through. So I spend the next 15 minutes or so on gene editing and digital agriculture. And we'll start by just giving an overview of. I'm sure many of you are familiar with this. I think this audience is very highly qualified. But what genome editing in a nutshell, differs from genetic modification in one sense, in that you don't leave any foreign DNA behind, and you're trying to mimic natural mutations. Now, there's been a lot of discussion on the types of editing that can be done, should be allowed, and without regulation. That conversation is still going on, unfortunately in India, but other places have made some decisions, other countries. Now, the goal is to, of course, be able to be in a position where your edit is equivalent to something that could be generated by chemical mutagenesis or other kinds of natural mutagenesis, natural or unnatural, whatever you call it. But if they need mimicking natural mutations. And so, there are options to be tried, talons, CRISPR. We have tried a number of these, and we've, as like many people, we've settled on CRISPR. So what I'll talk about is some of the work we've been doing. I always find it better to talk about actual work rather than just in general. So I wanted to give you a snapshot of what we've been doing the past few years. So we focused on CRISPR, but I'll show you one example of talon as well. And the flow is very straightforward. I'm sure many of you are familiar with it. So we design guide RNAs, make constructs, do regular agrobacterium mostly, mediated transformation. This is an example from rice, I guess, and generate transformants. And then the actual work starts later on, because you want to identify the mutants and get rid of Cas9 so that it doesn't keep introducing mutations. And that's what we've been doing in rice, in two crops. So I'll talk about tomato and rice. The first I'll talk about tomato, because some years back we published sort of proof of concept, because I wanted to be sure that we're doing it right. And many labs around the country are working on it. But we started very, at a very sort of low level, and used PDS, which is phytoene desaturase, just to knock it out and get albino phenotypes. So we were successful, and we got the mutations in the right exon that we were intending. And so this gave us a lot of confidence to go on to work more in tomato. So I should say that Myco has a breeding program in tomato, market leaders, but we do have a good set of germplasm. So one of the ideas was to, because tomato is a very good target for gene editing, there's lots of genome information, obviously, and there are labs around the world have done a lot of work in this crop. So what we wanted to do as a test case is to work with a gene called pectate lyase, and pectate works in the ripening pathway of the fruit. And essentially what our goal was to, number one, can we get mutations edit in a target region? And secondly, what are the phenotypes like? So we wanted to do a very close study on, actually linking the mutation to the phenotype. Because if we don't have that, the breeder can't use it. It has to be stable, inheritable, etc. All those things that are required by breeders. So we've worked quite a bit on generating a large number of PL mutants. And these are now ready in terms of characterization. So we've done, sequence characterization. We're doing off-target characterizations because that is deemed to be important. And I'll talk about regulations towards the end. But we can get much better shelf life, up to 30% better shelf life, better firmness in the fruit. All these things that you'd expect from knocking out this gene, and that was validated. So the challenge here is not to generate the mutation or get the plants. It is actually to get the right phenotype, because you need to balance, number one, the product characters. So whether it is the sugar content or the acidity or the firmness of the fruit, ultimately this goes in the market, and the consumer is going to choose it, or the farmer or whoever is in that chain is going to make the decision. If they don't like it, it's not going to make it. So we have to be extremely conscious of that, and then so we do a lot of screening at this stage to make sure that whatever we hand over to the breeder, it has to be absolutely usable in a commercial sense, as well as have that trait that we intended. Because otherwise, if you can do it conventionally, why bother? But we have to introduce something new, but have it fit into the commercial breeding perfectly. So that's the biggest challenge. Now, there are in tomato, there are many traits of interest, and these have been demonstrated in other labs, other companies as well. So this just gives a snapshot of things that are, one of the biggest things is nutritional value in tomato. Lycopene, if you can manipulate lycopene content, that has value. The Brix value, then disease resistance, color, all these things are amenable to manipulation by editing. It's a lot of work, but one by one you can actually target these. And the biggest things would be things like disease resistance. There are those many elite hybrids that may be lacking in, say, powdery mildew resistance, and you can mine the genome and get information and address those. So these are quite doable, I think, in tomato, crop like tomato, with so much genetic information and ease of transformation as well. I'll switch over to rice for a few slides. We started this back in 2017. We decided to work on rice, and mainly on two things. One is yield and producibility. That means because we work on hybrids, you want the hybrid, when the male and female lines are crossed, that producibility index should be high. It should be very efficient in producing seed at that level, because many good hybrids fail at that point. You're not able to produce enough seed from the male to female cross. So we were very fortunate to get BIRAC funding. They've been a great support. And what we did is we chose 3 or 4 genes, and I'll talk about them. The one that's shown here, IPA, is ideal plant architecture. It's a mutation which gives a change in plant architecture, which is more desirable from a growth standpoint, from a productivity standpoint. And we use talon for this. So this is a single example I'm just showing of talon, where we got a successful targeted edit in one single base pair change. But it took a lot of effort and it is expensive. So that's why we kind of prefer to do CRISPR-Cas, which is much easier and less resource intensive, which I'll show on the next few slides. So I'll show you 3 genes. One is, this is DEP, which is dense and erect panicles. And this is a very crowded slide. It just shows that so much work has been done on it. And these are from the literature. So we also generated new mutations in exon 5 and have characterized that very, very thoroughly now over about 3 seasons now. And I'll just skip ahead because so these are all the what is in the literature and what people have done and found using manipulations of these genes. So these are the 3 genes I'm going to talk about are straight knockouts because those are the simplest kind of editings that we can do. And we are restricting ourselves to the SDN1 type of mutation where we target very few base pair edits. And we see, if I go to the next slide. So on the bottom right-hand side, the plants are slightly dwarfed. So E stands for the edited plant. The plants are slightly dwarfed. But if you go on the left side, here we were very concerned about the grain quality, grain characters, and we found that the grain size is slightly smaller, but the yield has gone up by up to 50% in some lines. I'm not showing all the data because of time constraints, but we have a very good data set that shows that yield goes up by, at least if you average it out, the mean would be somewhere in the 20% range. And that's significant. Now the question is it, again, back to the question of, is it acceptable for commercial breeding? That's the big question. So we are now going to introgress this particular edit into our parental lines, the B line, A line, which is also a laborious process. So the male and female lines have to independently have it so that you get a homozygote in the hybrid. So that's where the effort goes in. So we've spent some time in generating these and validating it. So we know where the edit is precisely, we know the performance parameters. Now we need to see whether it breeds true and how does it breed in different genetic backgrounds. That is the question that we need to address. Second gene I'll talk about is DST, which is drought and salt tolerance. And the name suggests that it gives drought and salt tolerance, but that was not our primary interest. It was again in productivity, in yield. And there are 2 or 3 examples in the literature and showing increased grain yield. We've also targeted the same exon and got new mutations with increased grain yield up to 30%. Now, here again, if you look at the grain characters, they are not changed. One of the things we saw is on the left side, if you see the edited plants, and on the right is just a close-up of a leaf. You see that it's darker green in color and the leaves are slightly broader, so very easy to pick out the phenotype. The good thing was that the grain quality does not seem to be affected. But so these are the things that need to be addressed. So either you need to clean up that genetic background to get back to your perfect hybrid with the increased yield, and that takes a lot of breeding effort along with marker-assisted backcrossing and so on. The third gene I'll talk about is an interesting one to me, which is it's called QSTL3, and knocking it out actually increases stigma length. And the reason we did this was that one of the goals is to increase hybrid seed productivity. And so there was one example where this gene was looked at, and so we generated a mutation in the same exon. And interestingly enough, after a lot of screening, we could find lines which had increased stigma length. So the picture at the bottom left just shows the control versus the edit. Now I don't know whether the acid test of this does it really produce more seed in the final cross. So that we don't know yet. But it's encouraging to see that we can get multiple lines which have increased stigma length of varying differences, and the seed quality does not seem to be seed size at least does not seem to be affected. The plants look pretty much the same, edited or controlled. But it's very encouraging that we could get a specific change in the stigma length. So that's exciting for hybrid seed breeding. I think that needs to be checked. So the big sort of $1 million question is what can we do next with gene editing in India? So we're at a point where we're capable of doing many things in many crops. I think labs around the country are working on it and have reached quite an advanced stage in coming up with new phenotypes in different crops. The problem is on the regulation side. I remember I attended a meeting a year ago, more than March 2020 was when the gene editing draft guidelines were to be finalized. And suggestions were taken from all stakeholders, and I think there was a very concerted push by both public sector and private sector to make the regulations, categorize the regulations in such a way that the simple edits could have less sort of heavy regulations where products would come out faster without compromising any safety and so on. Unfortunately, it's now June 2021, and we have not seen the final regulations. We are hoping that we can get this done quickly, because there are so many things that are ready to go that can be field tested. Some of the examples I showed you, other labs, have many other companies are also working on it. So I think that's the next step for me, at least, is having the ability to take it out of the greenhouse. The other big thing I touched upon is, in all these crops, biotic stresses are becoming major problems, whether it's vegetables or row crops. And we need new tools to address these biotic stresses. Just for example, in rice, sheath blight, neck blast, these are a couple of examples where there is no real available tools in hand to address them directly. We can avoid it, but there's no real direct mitigation of the disease, tool to do that. And I think gene editing gives some avenues to test, potential genes for those capabilities. And I think if we miss out on this, it's going to be a big loss for agriculture. Okay, so the last part of my talk, I'll talk about digital tools and a couple of examples. Now, I'm not an expert on spectra, but essentially we are utilizing multispectral imaging to capture and get information out of plots. So what, in Myco, what we're doing in R&D is trying to assist the breeder in making better evaluations of their screening methods. So side by side with all the marker analysis, we can use digital tools to take pictures. Imaging is the biggest, one of the biggest tools here, is to take plot images and then analyze in various ways, and show a couple of examples, which will give the breeder some more information, some information that he would not have had earlier. And just to take some simple examples, NDVI is non-destructive vegetative index, and it's a simple tool of checking plant health at a very crude level. It checks the, it has it measures chlorophyll index in a way. And here, if you see on the left side is an irrigated plot, on the right side is a rain-fed plot. And this is very simplistic, of course. You're just seeing the reflectance, from the plants. But it is a tool that can be used to screen different genotypes. So if the genotype has a different reaction to water stress, if we can fine-tune that reading enough, we will be in a position to try and select plants based on their ability to withstand moisture stress. And here is an example from cotton. So if you look at growth, also NDVI readings across the growing cycle of the crop. So from vegetative stage on the left to boll bursting on the right, and each of those individual lines represents a single entry in the plot. So it's a large breeding plot. And what we are tracking is NDVI through the growing season. And as you can imagine, if you can track that index accurately, and given the plot has pretty much the same inputs as far as water, nutrients, crop management is concerned, but better performers can be selected out of such an analysis. And this is again very simplistic, because we are taking one parameter, NDVI, but you can also use other things like canopy temperature, and those sort of tools to judge, how plants react to moisture stress, temperature stress, and so on. There's a lot of excitement about early pest warning systems using this technology. So this is NDRE, which is non-destructive red edge. So it's a different wavelength. But we were trying to look for plots where you can see that there is some. It's a validation process. Again, a lot of these things are work in progress. So if you see a plot where you see early signs of some disease, in this case it was sucking pest. Can you take images early enough in the growing season, or before the pest is visible or pest damage is visible, and utilize that as a tool for taking measures in the plot. So this can be used by the breeder, by the farmer at some point. It's a very early stage kind of thing, because so many things can give you a differential reading. It could be moisture and water stress, soil conditions can give you different. So it's all up in the air right now, but we're trying to narrow it down to a point where we can validate this type of approach for a particular purpose, such as sucking pest detection. And I'll end my discussion with one example of a tool we're developing for breeders. This is a simple cotton boll count using digital tools, and the goal is to take an image with a drone on a cotton field, and then utilize that information using machine learning to estimate yield very correctly. So it has to be accurate. So the number of bolls has to be counted very accurately. We can then extrapolate the yield by using an average boll weight, which is known for that particular hybrid. So, that is simple enough. So essentially what we did was, I'll show you some pictures. We let the drone fly over, take pictures at high resolution, and then let the machine annotate the bolls, and then do what's called the ground truth confirmation. That is, go on the field and actually count individual bolls by hand. And so then compare the two and see how accurate you are. So we did this at different stages in crop growth, when the bolls are still green and then the bolls have opened up. So what I'm going to show you is actually at the later stage, because you have that contrast, white boll versus background, which is much easier for the machine to pick up as opposed to green boll. So I'm going to concentrate only on the late stage. This is 130 odd day crop. The bolls have just opened up, and what is shown here is one image. The red boxes are what was done by hand, annotated, and the green are the ones predicted by the machine. Now you can see that if you look at this one, it's an error, right? The machine has picked up a leaf on the ground thinking it's a boll. So that's where the error comes in, and we have to be very objective in saying how good is this tool, because ultimately, what is the use of this? It's going to be a time-saving device, number one, and it should be as accurate, if not more accurate, than manual counting. And here is another example of another plot with the same approach. Now, what we found is at this stage, where the bolls have opened up, we can easily get 96-97% accuracy. And you can train the machine as it goes through iterations. You can train the machine to avoid those false positives. So, for example, it's not picked up on the ground in this image. So that's where we feel this is one example. The other way to do things is phenotyping for the breeder. Develop a machine learning tool for measuring fruit, for example. So say okra or chili or brinjal, the major crops that we work with. We've developed a tool where a smartphone image used to measure the fruit length of a hybrid, girth or cross section, all these parameters, and take a mean of multiple replicates, and then export that to Excel and send that to the breeder, and integrate that into our breeding platform, phenome platform, so that all that data is in one place and it's instantaneous after taking the picture. So I think this is my last slide. I'll stop there, and hope there's some questions. The overall goal of all these efforts is to assist the breeder in reducing time, effort, and increasing accuracy. Thank you very much for that.
Sujata, you are going to take questions, right?
So my talk was crystal clear, no question.
No. I think there are many questions. I thought Professor Sujata is going to summarize some of these questions and give you the idea.
Okay, sure.
Yeah, many of those questions are regarding the position at Mahiko. If there are any positions available, are you going to hire anyone in Mahiko? That maybe we can start with that.
I'll answer that. So we're always I've given an idea of what we're kind of focusing on now in the recent years. Now there has been I think in general in industry, just to be open about it, is that there has been reduced hiring overall because of there's been a number of things happening on the technology front and difficulty in getting things through. For GM is one example only, I know, but in general it has cast a it has had a depressing effect on industry growth. I think from an R&D perspective, I'm talking about R&D perspective. Things have companies have become a lot more conservative in spending and hiring, so that has its knock-on effect. And I think I don't want to belabor the point, but I think if there's an enabling environment, I think more growth opportunities. So right now we're being specific to Mahiko, we would be looking at these specific areas I talked about, not in the wider sense, but anybody I'm happy to, if you contact me, I can give you feedback for sure.
Yeah. I think another question which is many of them have asked is regarding the genome editing technology. What is the future of this technology? Because in EU, in European Union, it is still considered as a GM crop. And if we look at our strict norms of GM crops, what do you think would be the future of gene editing?
So I'll talk about India, obviously. I'm optimistic that ultimately we will be able to do the SDN1, SDN2 type edits with reduced regulation. Now, what that means, I don't know exactly, because as I said, I had one slide on regulations with a lot of question marks. That needs to be decided. But I think seeing the opportunities that the technology gives, we would, both public sector and private sector have come together on this. And I'm very optimistic that at least the straightforward edits would have application with reduced regulations, and not the European model, I hope, for sure. But there is a great opportunity there, and I am very optimistic. It may take time, as these things do take time, but I think in this year or next year we should see some movement and ability to take things out of the lab.
Are there many private sectors interested in gene editing? Because earlier most of them were interested in the GM crop.
Yeah. So that, as I mentioned, a lot of the GM funding closed down. Many industries either put it aside or basically shut down their efforts in GM. Now, so it's a wait and watch thing. So it's, how can you, my personal view is that you need to be abreast of all these new technologies, be able to work with them, and then see how the regulations go. I think that would be my approach, because you can't be left behind. I think as an industry, we have to be active in it. So that's why we were very active in trying to get BIRAC support, other support to do this activity. Because frankly, management would be cautious on investing in something that may not pay out.
Yeah. I think Sujata is ready with the question.
Yeah.
Actually, you know how I'm getting muted.
Okay.
Anyway, I missed some of the questions. I think Vidya, you've asked all the questions which were.
No, I just talked to him about the gene editing, the regulatory issues, and what is the future of editing.
Okay, so there are some more questions which have been put up by the participants. One is that which genes would you target for editing for improving the photoinsensitive trait in crops, in flower crops?
Actually, we have not looked at that. That's not been one of our targets, but maybe offline we can take the discussion.
Okay. I think we have already told the participants to send their questions to us, and then we'll direct them to you so that you can answer them.
Yeah, sure. Yeah. I think that would be better.
So I think we should end the session now. And I thank Dr. Bharat Char for a wonderful session that he has given. He has given us a lot of information on all the forefront areas in this subject of plant breeding. And not only has he given a theoretical overview of the thing, but actually how it can be put into practice and develop products which are as a result of molecular breeding. So I thank you very heartily, Dr. Char.
Thank you.
And we will send you the questions later from the participants.
Yeah, absolutely.
Yeah.
Thank you.
Okay, so with this, we will end the session. Thank you.
Thanks, everyone.
For participating.
Thank you.